Hello, everyone. Welcome to the 2026 BCVS meeting being held here today at Boston. My name is Joe Wu. I'm a cardiologist at Stanford University. And with me today are very, very special guests and friends. We have Dr. Gordana from Columbia and also Dr. Aitor from MSU. And I'll have them briefly introduce themselves before we start to dive in.
Thank you, Joe. So it's really nice to be here today. My name is Gordana Vunjak-Novakovic. I'm university professor at Columbia, and I am directing a laboratory that is doing a lot of tissue engineering work between some regenerative medicine therapies and organs on chip that we are designing and using to model diseases.
Aitor.
Thank you, Joe. Thank you for having me here today. I'm Aitor Aguirre. I'm a university professor at Michigan State University. Our lab works fundamentally with cardiac organoid technologies and assembly technologies to create in vitro copies of the human heart.
Mhm. And so probably one of the most exciting things that happened past couple of years is back in April 2025, the FDA announced this new Modernization Act and also this NAM, so New Approach Methodologies. And this is followed by April 2025 as well by the NIH, in which NIH encouraged scientists to work on human-centric biology. And here we have probably one of the world's expert in this NAMS development for the past 20, 30 years. And so I'll start with you, Gordana. I want to get your take about the importance of this NAMS emphasis by both the NIH and the FDA.
This is really huge because it opens a possibility of using the technologies that we have developed to serve regenerative medicine for something completely different, which is to model human diseases and to test different kinds of therapies. So why is this important? In the first place, we always acknowledge that we have learned tremendously from animal studies. But animals have limitations. Human physiology and animal physiology may be very different. And I think cardiovascular disease is probably an area where this is very, very obvious. So if you can design physiological systems that we call microphysiological systems and alternative, I mean, there are many different names, but essentially you design functional units that consist either of one single tissue. For example, you can study heart muscle under different conditions, or you can link by vascular perfusion a couple different tissues that interact in the human body under certain conditions to model these conditions. It all started about 15 years ago or so. So NIH took initiative to actually ask the scientific and clinical community, can we design systems that have predictive power for like recapitulating some of the conditions in the human body? And if you can do that with the control that you can have in vitro, then we really have opened many, many opportunities. As you said, this evolved, so the organs on chip and organoids are more important today than ever before. And I think it is now on us to be good scientists, to really conduct basic science experiments and translational studies while constantly benchmarking and validating against human situation, so we can really distinguish what we can do and cannot do, where the limits are, but also where the possibilities are. And I just want to mention that it is not only FDA, it is also NIH that aligned with this Institute for Optimization of the Organoids. So they also invested into the organoid models. So, I mean, it's now up to us to see how much we can really do in this field to benefit patients and to benefit society at the end of the day.
Great, thank you. And Aitor, what is your take on the NAMS movement, as well as give the audience a sense of your history or your background coming into this field?
Yeah, so about the NAMS recent developments, I think that's great news. I think the NIH is doing the right move, basically. I think these models have been around for some time, right? Many of us were doing NAMs before the NIH announced these new initiatives. So it's a recognition that the field has been moving forward and that it's been impactful, right? And so I remember early papers, in the early 2010s, and many of them, by you, by Gordana, by many other people. And we have come a long way with these technologies. I think they are starting to reach what I would call critical mass, right? They are starting to become so good that in some instances they can actually substitute animal models, not for all purposes, but for some of them. And so I think if we continue putting effort in these technologies, they are going to continue, evolving and helping us substitute more animal. I don't think they are ever going to completely substitute animals, but I think they might help us push human-centric research, improving drug development, accelerating drug development, disease modeling, etc. So they are going to bring a lot of benefits to the cardiovascular field. My trajectory, it's a little bit unusual. I'm going to summarize that a lot. So I am a biochemist by training, and then a tissue engineer by further training. And then I became kind of a cardiac developmental biologist too. I was working in regeneration at the Salk Institute for my postdoc. One thing brought the other. iPSCs became so exciting at some point in the 2010s. And then I remember reading a lot of papers, many of them from your group, right, about iPSC disease modeling. And, like an idea started to, form in my mind that perhaps we could grow organoids from the heart with iPSCs. And so when I started my independent lab at MSU, this was one of the first things we tested. I was not sure it was going to work, but it did work, and here we are today, right? So now it's an exciting field. So
well, you've done tremendously well, and I think the field needs more people with a different training, different background, different perspective to come in to tackle different questions. Yeah.
I have to say you're absolutely right. Yeah. And if I could say something to the audience and to all the trainees out there, is that having a varied training and interdisciplinary training, it gives you a new perspective of how to solve problems. I think that's really important. We should emphasize that for new generations.
And Gordana, you trained so many of your trainees, and so many of them doing so well. What is your advice for some of the early trainees who are watching this video? What is it that they should do to prepare for this NAMS movement?
Yeah, I think there was never a better time to work in biomedical area. So I trained as a chemical engineer. I learned all my biology, medicine, everything that I needed to know, like a sort of on the go. What I would like to suggest to you to think about is, number one, follow big ideas. Follow something that's really, really hard to do, because you need to dream big. You will be working hard no matter what you take. So you better make some difference in the world. Find great people. You cannot do much alone. None of us can. Even a whole field cannot do too much alone. So surround yourself with very, very good people. I think that the progress that we are seeing today is to very large extent due to collaborations. So we collaborate more effectively than ever before, I think, in the history of science. So I think you should really ride on this way. Be brave and be persistent. Nothing works in the first try, very rarely. I believe that failure is the normal part of the process of discovery. When some of my students are having like a series of very successful experiments, then we are kind of, both of us become a little suspicious, like, are we innovating or not? Like, how come that everything is so smooth? And I think that the last thing I would say is what has changed in our field, and it's a very good thing, is that instead of this bench to bedside paradigm, like we discover something and we look if this can have application clinically, we actually flipped it. So you start your projects by talking to clinicians like Joseph Wu to understand what is the problem really, what is the critical problem, critical gap. And not only that, but how this needs to be addressed so that it fits into clinical flow. So like clinical need informing the opportunities that we follow.
Those are great answers, and I took some notes on myself and mentally as well. So I think Gordana talked about some big ideas. Maybe give some audience members who may be your future prospective at PhD or postdoc some of the big idea projects that you're working on. Obviously, your lab has had a series of very successful papers over the past 2 to 3 years, and that I've been a big fan of. Yeah.
You are putting me on the spot.
Yeah.
Some of the big ideas, I think some of them, I think many of them are kind of obvious, right? Like we need to prove more relevant physiology. And the way how we do that is by, developing more efficient vascularization, by developing I'm very enthusiastic about immunity. I want to see organoids with more immune properties. Mm-hmm. And also, we were discussing this before, Gordana and I, it's multi-tissue, right? It's, like tissues don't exist in isolation. So if we have a heart, that heart is receiving information also from the liver, from the lungs, from other parts of the body, from the brain. And so we need to start thinking about those multi-organ organoids. I still don't have a good name for them. Mm-hmm. Someone will probably come with a much better name for them. Mm-hmm. I think those are some of the big ideas that we need to start thinking about, because the field is getting ready for that. Right. That's my impression.
Can I add one thing? It just occurred to me. Everything we do, we should really try to do in a way that the therapy that we potentially develop is affordable. Because if you develop therapy that costs $1 million per patient, it will be limited to like a small cohort of people who can. So we started to think about it. So this is really a kind of economy of scale that is becoming very, very important in our field.
And so coming back to the question of affordable, some of the criticism or complaints by the trainees is that the MPS looks good, but it's so difficult and so costly to set it up in a small lab. How would you be able to say 10 years from now, 5 years from now, whatever that's being developed in both of your labs, and I'm looking at you, Gordana, you have some of those very beautiful multi-organ systems. It would be nice to be able to scale it up and to make it simple so that different labs can adopt. How are you thinking about passing this on to others?
We are really thinking about it. So this first original platform looked very pretty. We all loved it. But actually, there were just a few students in my lab who were able to make it and to run it. We put a lot of effort into streamlining it, simplifying it. And we also did another thing. I'll mention this in my talk today. Instead of doing the normal thing that our institutions expect from us, which is like before you submit your manuscript, you file IP, you file patent application. We just decided to bypass this and to publish the platform itself. It was heart-only platform, but with a lot of these small size heart muscles. So we had a platform and we had an actually AI software that is measuring contractile function in real time. So we just published everything open source, meaning every step, every detail is actually disclosed. People contacted us. We started to give these platforms to other labs, which was beneficial to them, but it was also very beneficial to us because we learned from other users what works, what doesn't, and how to, what you are describing, how to get there, how to democratize how to share knowledge, but also how to share hardware, because it makes no sense for us to put a lot of effort into developing something and then to keep it close to your chest instead of sharing with your. And we also do another thing, as much as practically, I mean, I'm sure we all do that, which is exchange of people. I really like to send my people to other labs and to receive people from other labs, and they work together under the same roof and figure out a lot of things, also from the practical domain.
No, those are very good points. And again, for the trainees who are watching, you're welcome to reach out to
Yes,
Gordana, for
absolutely.
help. And we've actually reached out to Gordana for help in the past, and she's been very kind and always willing to help us with any questions that we have.
And the
Yeah.
other way around, because we have a ton of cell lines that your lab developed that you very generously give to others. So
Yeah,
I
good,
think
good.
that
And then Aitor, so maybe I want to ask you that you mentioned that in the future, you don't think you'll ever replace mice. And I think there's some misperception among the scientific community that NAMS is over there to take over animal work. So I want you to clarify that to the audience and members, that it's more of a synergistic collaboration rather than one technology replacing another technology.
Yeah,
Maybe you could,
absolutely. I think, NAMS is another tool to add to our repertoire of tools, right? And it can be a very useful tool, especially in combination with animal models. I don't think NAMS are going to substitute animal models anytime soon, perhaps in a very long, very, very long time, but I won't see that. I think, it's another tool is like, for example, cell culture was a tool, right, that was developed in the mid 20th century. And now every lab has a cell culture, or almost every lab has cell culture capabilities. So I think NAMs are that. They are a tool that we are adding, and in some spaces it might be more useful and might substitute more. In other spaces, might substitute less. But it's still, the animals are still there, and they are still going to be there for a long time. There's also a lot of animal models that have special qualities or properties that make them not possible to substitute, right? Because they have a particular property in their heart or in their physiology that is very interesting to study, I think. Finally, I would also say, and I've been spearheading this, at least fighting for this idea for years now, that there is also a space for hybrid models where you have an animal model that contains human tissue implanted in it, right? So our organoids, we are implanting them in mice, and then we are following the mouse. The mouse is kind of like a living bioreactor that is maintaining that human tissue alive. And so we can study human physiology inside of a mouse, right? And we still have all the mouse cardiovascular system to use as a control, as you see the differences. I think these hybrid models are going to become more and more popular as we move forward, right? So I think both systems are going to coexist. Yeah, that's my.
Good. I think that's a good message for the audience to know that it's actually a synergistic collaboration, not one technique replacing the other technique, because ultimately you want to use all the techniques out there, as Gordana mentioned, to come up with therapeutics that are cheaper, faster, that we can deploy in our patients as soon as possible. Both of them are going to give 2 outstanding talks in the upcoming hour, and I want each one of them to give you a sneak preview of what they will be talking about. Gordana, start.
Okay, so I'll talk specifically about organs on chip for cardiovascular research. So I'll tell a little bit about how we conceptualize them, how we design them, what is important. And I will speak about the use of just like a heart muscle alone model, very simple model that we use to model different kinds of conditions that are seen in patients from say, genetic cardiomyopathies to ischemia reperfusion injury to, like, for example, autoimmune disease caused cardiomyopathy. So I will talk about this, and then I will close with a little bit of perspective about like where we are now and where do we go in the coming years, because I think the future is really very, very bright. We should keep doing what we are doing now, which means like collaborating within our community and being mindful about how to invest our time. And also being mindful, so this is now about you, I think that mentoring young talent is the most important and the most rewarding part of our job. I think the three of us will agree about this, and modalities for training are changing as the field is changing. So we kind of grow together. because you are taking over the torch. I mean, we will continue working, but not forever. So for us, it's very, very important that you are enabled not only to learn what we know, but actually to develop ability for critical thinking, for collaboration, to be ready for the future, which is obviously very unknown to all of us.
And Aitor, your sneak preview as well as your positive message to the trainees.
Yeah, my sneak preview. I'm going to be talking about the work that we have been doing over the last 5 years on heart organoids. I'm going to be talking a little bit about development of the heart that we have been studying those systems. I'm going to be talking a little bit about disease models, and I'm going to be talking a little bit about applications, in the space of cardiac safety, cardiac toxicology. And I will give a sneak peek about what I think are some of the next generation models, more assembloid type work. And so, yeah, that's a little bit what I'm going to be talking about. And my message is, well, Gordana was mentioning it before, emphasis on the trainees. Think big, because if you think big, you might not get big, but at least you have that potential. If you think small, you will stay small, even if you meet your goals, right? So for the trainees, is think big, be faithful to your ideas and what you believe in, and it's tenacity, it's continue insisting, even if people are telling you that's not going to work. You believe in it, just stay true to it, because sometimes you reap the benefit. So yeah, that's my message for the trainees.
Oh, good. So I want to thank the audience for watching this session here. And I also want to thank Gordana and Aitor, really two of the ones that leading expert in NAMs. And for those of you who don't know what NAMs is, I hope at the end of the session you know what NAMs is. And I hope that you would open up your mind and think about diving into this topic. And as Gordana and Aitor both mentioned, that we're very happy to collaborate with any investigators who's interested in using these human-centric biology to come up with novel therapeutics in the future. Thank you very much, and bye-bye.